Ever wondered what makes you you? Like why you have your mom's eyes or why certain things just seem to run in your family? Well, today we're diving deep. We are going to crack the code of life itself and start to uncover the amazing genetic story that's written inside every single one of us.
You know, this whole thing for me kind of started with a super simple classroom experiment. Maybe you've even done this one yourself. You get this little strip of paper with a chemical called PTC on it, and you stick it on your tongue. For some people, it's just wildly bitter. But for others, like the Amoeba sisters and, yep, me too, it tastes like, well, paper. Absolutely nothing.
And right there, you've got a fascinating puzzle, right? How can two people have such a completely different experience with the exact same thing? And it's not about being a picky eater or anything like that. It is a genuine biological mystery. And it's a mystery we're about to solve.
Turns out, the answer isn't on our tongue. The secret is hidden way deeper inside our cells, woven into the very molecule that carries the instructions for, well, everything our bodies do, our DNA. So, to really get this tasting mystery, we first need to understand how our body's instruction book is organized. And there's a great way to think about this. Imagine all of your genetic information is like a massive library. That entire library, every single piece of data that makes you who you are, that's called your genome. It's the complete set of instructions for building and running you. Now, a library isn't just one giant, endless scroll, right? It's organized into books. In our cells, these books are called chromosomes. We humans have 23 pairs of them, and for each pair, you get one book from one parent and the other from your other parent. And if we zoom in even further, each of those books is made up of chapters. These chapters are our genes. A gene is just a specific section of DNA that has the instructions for one particular thing, like the gene that's involved in tasting PTC. And finally, what are those chapters written with? Letters. The absolute most basic building block, the individual letter of our genetic code, is a chemical called a nucleotide. Millions and millions of these letters, in a very specific order, are what make up our genes. So what do these letters actually look like? Well, DNA itself is a double helix, which basically looks like a twisted ladder. The rungs of that ladder are made from pairs of these nucleotide bases. And here's the magic part. There's a strict rule. Adenine, or A, always pairs with thymine, T. And cytosine, C, always pairs with guanine, G. This A to T, C to G pairing rule is literally fundamental to how all of life works. Okay, so we've got the instruction book. We know it's written in an alphabet of 4 letters. But how does the body decide which instructions to follow? For that, we need to get into the rules of the game, genetic inheritance. So remember, you get a set of chromosomes from each parent. That means for every single gene, you've actually got 2 versions. These different versions of the same gene are called alleles. You can think of it like having 2 slightly different recipes for the same cake, one from each parent. Now, some of these alleles are what we call dominant. We usually write these with a capital letter. For a dominant trait to show up, you only need 1 copy of that allele. It's kind of the loud talker at the party. In our mystery, the ability to taste PTC is a dominant trait, so we'll call it a capital T. Other alleles are recessive, and we use a lowercase letter for those. A recessive trait, it's kind of shy. It only gets to show itself if there are no dominant alleles around to overshadow it. So you need 2 copies of the recessive allele. To be a non-taster, you need 2 copies of the lowercase t allele. And this chart shows it perfectly. Look at the dominant trait. See how it can show up in every single generation? If a kid has it, at least one of their parents had to have it. But then look at the recessive trait on the right. It can totally disappear for a generation and then, bam, pop right back up. The parents can be carrying the trait without you even knowing it. Okay, this brings us to 2 really important words. Your genotype, that's your actual genetic code, the combination of letters you have, like big T or big T little t. Then there's your phenotype. That's the physical thing you can actually see. Are you a taster or a non-taster? Genotype is the code, phenotype is the result. And to get a little more specific about the genotype, we use 2 more terms. If your 2 alleles are the same, like 2 big T's or 2 little t's, you're homozygous. Homo just means same. And if your 2 alleles are different, like 1 big T and 1 little t, you're heterozygous. Hetero means different. This difference is everything when it comes to predicting traits. All right, we are armed with all this new language. We know about alleles and genotypes. We can finally go back to our PTC taste test and solve the mystery. How can 2 parents who can taste it have a child who can't? All we need is the right tool. And that amazing tool is called a Punnett square. It's this beautifully simple little chart that lets us map out all the possible combinations of alleles the parents could pass on, and it even helps us figure out the probability of each outcome. It's a genetic prediction machine. And making one is super easy. First, you figure out the parents' genotypes. Then, you put one parent's 2 alleles across the top of the box and the other parent's 2 alleles down the side. Then you just fill in the grid, combining the letters from the top and the side. That's it.
So here's the key. For 2 tasting parents to have a non-tasting child, they both have to be heterozygous, big T, little t. They can both taste it, but they each carry that silent, recessive non-taster allele. So when we cross them in the Punnett square, look what happens. There's a 25% chance of a TT taster, a 50% chance of a TT taster, and there it is, a 25% chance of a TT non-taster. And just like that, mystery solved.
Punnett squares are awesome for predicting one generation. But if you want to track a trait through a whole family history, scientists use something called a pedigree. It's pretty much a special family tree that uses symbols to map out who has a trait, who doesn't, and who's a carrier, all the way down the line.
Okay, look, this is about way more than just tasting a piece of paper or figuring out if your earlobes are attached or detached. Understanding our own genetic code has some seriously profound implications for our health and our lives.
All of these ideas, dominant and recessive traits, pedigrees, probabilities, these are the actual everyday tools that genetic counselors use. They use this science to help real families understand their risks for inherited diseases and make really important, informed decisions about their own health.
And that's why this really matters. You know, as genetic counselor Jacqueline Haven puts it, it's a huge privilege to help families navigate these incredibly complex and personal situations. Cracking our genetic code isn't just a science experiment. It's a deeply human journey. It empowers us to understand our past and even gives us some tools to help shape our future. So, what story is written in your code?